this is probably by far the most important slide as far as understanding enzyme regulation goes, okay? So I want you to pay very close attention to this slide. You really need to know what each type of enzyme regulation entails, what it means, and also know the terms used for that type of enzyme regulation. So enzymes are regulated by a multitude of ways, distinct ways. What does that mean? That means you can activate an enzyme, you can inhibit an enzyme, you can regulate how much of that enzyme protein the cell produces. So this thing doesn't sit very well on my head. Okay, so we're going to go through the different types of enzyme regulation. The first one called, and I'll go through examples later on, but I'll give you an overview here. First one is called proteolytic cleavage to activate. That means an enzyme is produced as a zymogen, called zymogen. It's produced in an inactive form. The way you activate it is you truncate or clip off a short segment from the amino terminus of that zymogen to convert it into the active enzyme. In fact, most of our proteases in the small intestine are released that way, as zymogens. Inactive, once they hit the small intestine lumen, they're activated in situ, and they are able to digest the dietary proteins. Okay, very important. We'll give you an example in a little bit. The next kind of enzyme regulation is called covalent modification. And that's also extremely, extremely important. Half of the enzymes we cover are subject to covalent modification. So I want you to understand the term. The reason it's called covalent modification, you are creating a new covalent bond to the enzyme. Typically, that covalent bond, the most common type, is the addition of a phosphate. That phosphate typically comes from ATP, and you need an enzyme to add that. So you need a protein kinase. Okay, that's the class of enzymes I mentioned in the beginning of this chapter. Okay, so I want you to think about you're taking an enzyme, you're going to modify the R groups of the amino acids in that enzyme. What are the R groups that can be modified? Let's say that you can add phosphate to. Okay, good, nice. Okay, good. So hydroxyl group, serine can become phosphoserine. Threonine can become threonine or tyrosine. The outcome of that typically can be either to activate an enzyme, or depending on the identity of the enzyme, it can inhibit certain enzymes. So covalent modification is extremely important regulatory mechanism. And the most common one is phosphorylation. There are other forms of covalent modification. That means you add a different group. But phosphorylation is the most, one of the most important ones. Now, the reverse is true. If an enzyme is in the phosphostate, phosphoserine, and you remove that phosphate, you are breaking a covalent bond. So that's still called covalent modification. Okay? So either way, it's called covalent, whether you're adding a phosphate or removing a phosphate. Okay, third one is pretty easy. Sequestration. Some enzymes maybe exist as a single subunit or multiple enzyme molecules forming a polymer. In some cases, when you form the polymer, the enzyme becomes inactive. So that is called sequestration. You put many enzyme molecules together, so individually these enzymes become inactive. The fourth type here, allosteric regulation, is absolutely very important. Allosteric regulation is where a small molecule, typically again organic molecule, that is distinct, different from the substrate for that enzyme, binds to the enzyme at a site distinct from the binding site, the substrate binding site. It's called other site. And that's the term, what the term allosteric stands for. A small molecule binding to another site on the enzyme besides the enzyme active site, and changing the activity of that enzyme, either positively or negatively, activating or inhibiting. And that's again a very important regulatory mechanism. So I really want you to compartmentalize each of these ways. Don't mix them up. There's no one is covalent modifications is one way. Allosteric is another way. Don't mix them up. You want to make sure you know what they mean. So we'll cover some of that, some examples also with allosteric regulation. Now, the last two are kind of related. The last two, I'll start with the first one, and that's called, again, pay attention to the term, induction or upregulation. By definition, what that means is you increase the number of enzyme molecules in the cell. How do you increase the number of enzyme molecules in the cell? By increasing the expression of the gene that encodes that enzyme. That's the only way, that's the only thing that induction and upregulation means. So, for example, let's say the liver under specific conditions has two molecules of an enzyme called glucokinase. Under another condition, the liver upregulates the gene for glucokinase and makes 5 copies instead of 2 copies of that enzyme. So each cell will have 5 instead of 2. That's what you call upregulation or induction. When you do that, you're not necessarily activating the enzyme, but you're providing the cell with more enzyme. That means there's increase in overall enzyme activity. Okay? So this is very important. Now down here, this is called repression, which is the reverse. That means under certain circumstances, the cells may decide to downregulate the expression of a specific enzyme. We don't need that enzyme, so the cell doesn't produce that enzyme. Okay, or reduces the amount of that enzyme. That's specifically called repression or downregulation. Okay? These last two I want you to pay attention to are the slowest of all enzyme regulatory mechanisms. The reason they are slower, because obviously they involve multiple steps. You have gene expression. That means you have to transcribe the gene. Then the transcribed gene is going to be translated. The messenger RNA is going to be translated into protein. The protein is going to be synthesized in the ER and it's going to be modified. So all of those steps take more time. So these last two usually are the slowest, meaning they can take several hours, in some cases days. Okay, so this is the first one we talked about, proteolytic cleavage to activate. So there are many zymogens produced from the pancreas that go into the small intestine that actually allow us to digest protein that we take in, that we eat through the diet. So example is here, trypsinogen is a zymogen. Again, zymogen because it's in this form, it's inactive. When it's released, it's inactive. When it gets to the small intestine lumen, it travels from the pancreatic duct makes it to the small intestine lumen. Here, you see you remove 6 amino acids. You truncate 6 amino acids from the amino terminus. Again, every polypeptide is going to have amino terminus, carboxy terminus. So you remove 6 from the amino terminus. The enzyme is converted from trypsinogen, inactive, to trypsin. That's the active form. There are similar other zymogens released. The chymotrypsinogen is another one, and so on. Again, don't memorize the details, just understand the principle. You truncate a segment of the amino acid to convert the zymogen to the active enzyme. Covalent modification. Actually, it's really pretty straightforward. It looks like most of you have kind of tuned into that. So let's say you have an enzyme. It has amino acids that have hydroxy groups. Let's say it has a serine. That serine, therefore, can be modified. It's still part of the enzyme. It's not. So when you modify that serine to phosphoserine, maybe that phosphoserine is near the active site. Maybe it allows the active site to associate with the substrates. We don't know, but the point is you change the activity of the enzyme. So that's an example of covalent modification. Typically, covalent modification requires one of 2 enzymes to do that. So this protein kinase is the enzyme that adds the phosphate from ATP to this other enzyme. So this would be the effector enzyme here. And this is the protein kinase that modifies that effector enzyme. Okay? Similarly, you can remove that phosphate by a phosphoprotein phosphatase, bringing the enzyme back to the original form. So you will find out that there are many different types of enzymes that get activated or inhibited that way. So this is why this is an important thing to understand. Okay. So this is really just to tell you there are many different kinds of covalent modification. The most prominent one is here, enzyme. You have a phosphate added, typically on serine, threonine, or tyrosine. Those are the most common ones. Okay, you can add other groups, in this case bigger groups, but we're not going to get into them. But that's also called covalent modification because you are creating a bond with an adenylate group or uridylate group and so on. Third one is allosteric regulation. And this is really where I want you to really appreciate the concept of complex enzymes. Complex enzymes typically, as I mentioned, will contain more than one subunit. In this case, the catalytic subunit is designated C. Here's one polypeptide. Here's another polypeptide. The functional enzyme is both together. Okay? So this enzyme is made of two subunits. Now only C, the catalytic subunit, is involved in the catalysis of that reaction. R, the regulatory subunit, can regulate the activity of C. So here comes, so I want you to pay attention to two things here. S is the substrate. So the substrate eventually is going to bind to the catalytic subunit, where it will be changed to a product. M is an example of allosteric modulator. It can be positive or it can be negative. In this case, it's positive. So here's M, again, small organic molecule, different than the substrate, completely different than the substrate. Okay? It's going to bind to the regulatory subunit. And by binding to the regulatory subunit, it's going to activate the catalytic subunit. So the catalytic subunit can then catalyze or catalyze the conversion of the substrate to product. You can have a reverse modulator, negative, where it binds to the regulatory subunit and it inhibits the catalytic subunit. Okay? So this is very important to appreciate. Again, these are typically organic molecules that bind at a different site than the active site of the catalytic enzyme. Fourth type of enzyme regulation, fourth and fifth really, but I will talk about the fourth one because they're similar but up in the opposite direction, or do the opposite thing. So enzyme induction or upregulation. This does not mean enzyme activation. Okay? This simply means induction or upregulation, meaning increasing the number of enzyme molecules. So don't use this as synonymous with activation. It's not. Okay, very important. So this specifically refers to increasing the number of enzyme molecules. And I'm going to give you a good example, a real life example. Steroids, that's how steroid hormones work. Steroids, here's a steroid hormone. Let's say this is cortisol, important steroid hormone. It's going to cross the membrane because of its lipophilic nature, is able to cross the membrane. Here it is inside the cytoplasm. And here's its receptor. The receptor is protein in nature. Small protein receptor. So you have hormone, receptor come together. This is the active complex of steroid hormone and its receptor. The complex now can translocate into the nucleus, cross the nuclear membrane. It is going to find a specific gene. Here's the gene, the DNA, and it's going to bind, that complex will bind to a specific region in that gene designated enhancer region. When this complex binds to that region, it activates the promoter region of that gene, which results in increased transcription of that gene. Increased transcription, that means increased messenger RNA is made, more messenger RNA is made. If you have more messenger RNA, you're going to translate that messenger RNA into more protein. So steroid hormones, cortisol in particular, that's how it exerts its effect. It's going to upregulate certain genes, upregulate certain enzymes, increase the amount of certain enzymes. Okay? In some cases, you have the reverse of this effect. The reverse of this effect would be to have similar molecules, but binding to a repressor region instead of an enhancer region of a gene, reducing or repressing or down-regulating that gene. Okay, so as I mentioned, this is the slowest of all enzyme regulatory mechanisms. So this is really kind of a quick summary, but don't worry too much about this middle part. Just allosteric regulation, covalent modification are pretty fast, like happens within minutes. The last one is typically upregulation, downregulation, takes hours to days. Okay?